Hi, this is Deb, and we're going to walk through Chapter 6. I want to give you a heads up. You may have already looked at your book. This is a lot of chemistry. We're talking photosynthesis, and it can be a little confusing. So what I'm going to do in this video, and what I encourage you to do in the PowerPoint, is to just look at the diagrams. Walk through the diagrams. You can find a lot of answers by just understanding what's happening in the diagrams. And so that's what I'm going to talk about pretty much this whole time. All right, so let's see about what this generally is. This is something that you've heard about since grade school. It's how plants take energy from the sun and turn it into carbohydrates. Pretty seriously important. Nothing would be alive without it because that's our food source. We can't actually source out the energies from the sun and make our own food. We count on these plants to do that. So who can do it? Not only plants, but algae, some protists can, and cyanobacteria. But the bulk of it, plants and algae right there. Here's more examples of them. Kelp. Diatoms are these little tiny microscopic aquatic things that are so cool. They're actually, these shells are made out of glass. They're tiny. Cyanobacteria, Euglena, another little guy that swims in the water that you probably swim in yourself. You didn't know he was in there. It's very tiny. Plants and mosses. So, flowering plants. Here we got the same picture again. We got sunlight coming in. We need carbon dioxide and we need water. And what comes out? Oxygen. That's really just a byproduct. That's not the purpose of photosynthesis. This is the purpose of photosynthesis, glucose. So these go in, this comes out. Let's see. So what the main thing we're shooting for here is glucose. And I have another slide that talks about carbon dioxide being reduced better on another slide, but carbon dioxide is the building block that we reduce and turn into, say, glucose. Same, that's made out of carbon, oxygen, and hydrogen. So glucose is the main thing that we produce. If we look at the leaf of a C3 plant, which is the main plants that you see around you, all of this part here we'll see. All of this is performing photosynthesis or involved in it. The only thing that's not are these little outside edges. And here's the opening that lets the CO2 come in and the oxygen come out. On the bottom of the leaf, pretty smart. So if it rains, it doesn't go in. Kind of cool. All right, so let's look at a chloroplast. It's a double membrane, and it forms thylakoids in there. And so a thylakoid are these stacks of green things. They're made out of this thylakoid membrane. And the space is the innermost compartment of the chloroplast. And so we're looking at that. Let's see. Chlorophyll, when you look at a plant cell under the microscope, all the little green dots you see are the chloroplasts because they're green because of the chlorophyll. There's other pigments in there too, and we'll talk about what they do, but those are all in the thylakoid membrane, and so this is colored green to show that those are all in those membranes. All right, so we've got a chloroplast. Here's another picture of one. The stroma is the gooey stuff around them. Granum or grana is this stack of thylakoids. This thylakoid membrane goes to a stack, and so this is a whole system here to do this. The stroma is this thick fluid inside here, and it's really rich in enzymes because you've got a lot of reactions going on here, and these enzymes have to keep it going. Just another picture. This is what it actually looks like, a cell of a plant under the microscope. And you'll see all these little green guys floating around. They actually kind of move. This cytoplasm in particular flows. It's kind of cool under the microscope. And just another, you see the stroma, the gooey stuff is this space around here, and grana, these stacks of the thylakoid membranes. All right, here's where we talk about reduction and oxidation a little bit. So we start with, here's the equation. We start with water and carbon dioxide. And through this process, and solar energy, can't do it without it, and through this process of photosynthesis, we end up with a carbohydrate and a byproduct of oxygen. CH2 is often used to represent carbohydrates because that's the ratio. For one carbon, there's twice as many hydrogen and the same number of oxygen. So glucose is C6H12O2. This is just a formula that means it's that same ratio, and it could be any carbohydrate. All right? Okay, so let's look at this diagram at the bottom. I hope you can see it all. You can learn a lot from this diagram right here. Okay, first of all, we can see that CO2, carbon dioxide, is reduced into the carbohydrate, whatever that carbohydrate is. We also see that water is oxidized into oxygen. All right? So we got that part going on. Now let's look at the color of those. And so we see that the hydrogens, this water molecule is split. We'll see the actual, you'll probably see more than you ever wanted to see about that coming up. But this water molecule is split. And the oxygen goes over here into this byproduct of water. And we use those hydrogens to build, or the plants use the hydrogens to help build that carbohydrate. Because carbon dioxide only donates carbon and oxygen. And we have to have that hydrogen too. So, awesome little example there. All right. And that's called a redox reaction, reduction oxidation. So reduction here means a gain of electrons. Also, you can see it from here. And oxidation means a loss of electrons. So it's kind of, and then you couple them together. So that's a pretty common thing. You'll hear about redox reactions a lot. All right. There's two main types of reactions. There's the light reactions and the Calvin cycle reactions in photosynthesis. The light reactions occur in the thylakoid membrane. We talked about that. There's chlorophyll, blah, blah, blah. But all this is really doing is it's capturing the solar energy from the sun and putting it into a form that we can use it, which happens to be ATP and NADPH. So we capture the energy in the light reactions, capture light energy, put it in these two molecules, and then it goes to the Calvin cycle, where that energy is used to produce a carbohydrate, to take that carbon dioxide and the hydrogens from that water and turn it into a carbohydrate. So they have to take energy right here to build a carbohydrate, to store the energy in a better form. All right, so let's look at a bigger picture of this. All right, so we've got the light reactions here, and we'll learn that there's one and two. There's two different ones. Solar energy is coming in, and we're really talking about these little teeny green chloroplasts right there. All right, so light reaction. Water comes in, solar energy. Follow the arrows. What comes out? That's oxygen byproduct. And we're capturing, these get energized, and we capture that energy in an NADPH and an ATP. Then it swings over here where carbon dioxide comes in, and through the Calvin cycle, it grabs this energy off of those. Remember that phosphate pops off easily and releases energy? And it grabs that and builds a carbohydrate. Oops, which you can't see, I'm afraid. That's below the thing. I'm sorry. So carbohydrate is at the end of this arrow right here. So then these two molecules are recycled. They don't have energy now. They go back over here. If they get energized, pick up that energy, come over here, drop it off, build a carbohydrate. Back over here, jazzed up again, back over here, drop off of that energy. So it goes, you go through that, or plants go through that cycle thousands and thousands of times. All right. So let's talk about this. There's certain pigments that absorb solar energy. And so let's talk about solar energy. There's different wavelengths, and different wavelengths have a different amount of energy associated with them. There's visible light, which you can see, and we found that shorter wavelengths contain more energy and longer wavelengths contain less energy. Vision and photosynthesis are adapted to use the most prevalent wavelengths of what comes through and hits the Earth from the sun. Only about 42% of the solar radiation reaching the Earth actually gets all the way down to the surface. All right? So let's look at those wavelengths. So we have visible light. Goes from this kind of purplish through blue, through green, through yellow, all the way over to red. Got it? And that's visible light, right? So we need photosynthetic pigments that actually capture that light very, very, very efficiently. Turns out the chlorophyll, the green stuff in plants, there's a chlorophyll A and a chlorophyll B, absorb, look at this, there's A, here's B. they absorb a whole bunch of energy down here where there's lots of energy. This is absorption of light. How much energy is absorbed by different color wavelengths of these different chlorophylls and carotenoids. So we're talking about chlorophyll A and chlorophyll B actually being the green in the leaf. And these are the colors left over that you only see in the fall when the chlorophyll's gone, when the leaves turn orange and red and that kind of stuff. So these guys absorb most of the energy down here where those wavelengths are short and have a lot of energy. So they're much, much, much more efficient. And there's a little bit going on over here in the red. Nothing here through the green. Well, guess what? Green's reflected. Whatever color something is that you see with your eyes, that means those wavelengths are being reflected back and not being absorbed. That's why green leaves are green, because it can absorb all of this, and reflect back the green. I know it sounds a little bit crazy, but it is true. All right, so we have these guys here become the oranges and the yellows and reds that you see in the fall because the tree stops producing so much chlorophyll, let's say, so there's no more green showing up, and then we see what was there all along, these guys. And so they still pick up more of these wavelengths and absorb energy. Not as much as that, but they do still do it when they need it, okay? And so they're reflecting these yellow reds. Do you see this? It goes down to nothing. They are reflecting this yellow through red, not absorbing that. And here's another picture that shows this. Okay, so we have all the spectrum of lights coming down, light waves, and all of it's being absorbed by this leaf to take that energy and turn it into carbohydrates, except the color it is. That's what's bouncing back, reflected. And so green is reflected, and because green actually is the wavelength that does not have that much energy in it. Pretty cool plan. And so in the fall, what we see reflected back are the ones that aren't absorbing any energy. The chlorophyll's gone, and now these yellow to orange leaves are doing their thing. All right, so what exactly is happening there? Well, there's an electron pathway. So what we're really doing is capturing the sun's energy and storing it in the form of a hydrogen ion. So we're taking that water right there and we're splitting it. This oxygen comes off, and that's going to be the byproduct that turns into oxygen gas because we want all those hydrogen ions. All of this is happening inside the thylakoid, and this is the thylakoid membrane. We're going to find out that there's some photosystems coming up here that have this electron transport chain hooked up with them. But the end product of this whole thing is to take the energy from this and put it into this molecule, and to take energy here and put it into this molecule. These are the two guys. That's the whole purpose of that light reaction, is produce this energy that will then be used to build carbohydrates in the Calvin cycle. Right there. That's it. Purpose clear. All right, let's see it again. All right, we got solar energy coming in, water, we split that. The oxygen's the byproduct. Here are all those light reactions happening in the thylakoid membranes, and they're capturing that energy and putting it into these two molecules that go over here. The CO2 used with it to make a carbohydrate. Then they go back over here and get charged up again. All right, so we've got two photosystems. They're named for the order they were discovered, not in the order they go. They're photosystem 1, photosystem 2. Kind of crazy, actually, because they don't really go that way, but nonetheless. They consist of pigment complex and an electron acceptor. We'll look at some pictures in a second. That means that this electron is going to bounce along like a bunch of people handing down in a long line, passing a bucket of water to put on a fire. That's kind of how that electron does work. Let's see. And so we have the whole thing gathering the solar energy and passing it to the reaction center, which is a chlorophyll molecule. So generally, they just capture the energy of the sun and change it into a form we can use. That's bottom line it. So here's how the whole system goes. We've got photosystem 2, and those electrons are being energized from the sun's energy. We split the water, oxygen gas is released, and then they go through an electron transport chain. That's kind of like a bunch of people in a row handing each other a bucket of water to get it down to the end. They bounce these little electrons along. And as they do, they release little bits of energy, and they keep that energy and pass it down. And finally, at the end, we get the big payoff. Then you go to photosystem 1. Remember, these were named in the order they were discovered, not in the order that they work. So then we also energize electrons, and this part goes and puts that same energy into NADPH. So here we are making ATP and NADPH, the two things we really, really need. All right, so let's look at them. So we've got solar energy coming in here, and energy level goes up. As those electrons move along farther away, they get lots of energy in them, farther away from the nucleus. So water splits. Hydrogen's going to go over here and do something. Oxygen goes off to become oxygen gas, a byproduct. These electrons go down here and become part of this electron transport chain. Boom, it happens again. There you go. Check it out. So we have energy coming in through here. We save those electron transport chains, put that into ATP. Boom, over here. Then we do it again over here, and this one with the hydrogen ions that came off of it, that creates an NADPH. And both of these feed into here for energy to build that carbohydrate right there. That's the whole purpose of it. And this is how it happens on a, you can see it fitting into that. All of this is in that light reaction right there. Okay. All right, so let's look at the organization of that thylakoid membrane. This looks crazy. Don't pay attention to all the little details. We don't need those. What we really want to look at, that there's an efficient transfer of electrons, and we have hydrogen ions flowing along. That's all we're talking. So we split that water. Hydrogen ions, boom, and they go through here, through this kind of a turbine, which we'll talk about, the ATP synthase, the enzyme that helps build ATP. Boom, we now have ATP, and that hydrogen ion drove that. On this part here, we have electron transport chain, boom, 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 those electrons booming along, boom. We have more light going in, this is the PS1. And finally, they capture that energy and put it into NADPH. That's all I want you to see. Kind of crazy for that, isn't it? All right, so ATP production is this stuff in the thylakoid membrane. This whole thylakoid space, excuse me. This whole part here is a thylakoid space, and that's where these hydrogen ions hang out. And so they come here and they shoot through because they get really strong, built up here. They want to move to a lower area of concentration. They follow that concentration gradient, and that turns this turbine, which then captures that energy and puts that on that ATP. Pretty darn cool. Then what happens here, electrons are passed from one chlorophyll molecule to another, like this, until they ultimately end up in this whole electron transport chain. See this whole with these E's bouncing along into one central chlorophyll A molecule. That's going to like organize it. You'll want to watch that one on your own. All right, so we have that hydrogen gradient produces the ATP. Then NADP is something a little bit different. It's a coenzyme. It accepts electrons from that long electron transport chain, and it gets another hydrogen ion, by the way, through that whole thing. And then that is one form of storing that energy. So what's a coenzyme? It's a non-protein helper that works with an enzyme. So those enzymes that go through that make these reactions happen at the rate we need them to, those are proteins. But this coenzyme is not a protein, but it still does that kind of thing. Have that kind of job. So, just what I said before, if you think of ATP synthase as the complex as a turbine, when the hydrogen ions pass through there, it turns a turbine and the energy is transferred into an ATP molecule. Boom-o, right there. Okay, let's put this in kind of the big picture. See this whole little thylakoid membrane that makes these stacks, this is one of them. And so we have the water splitting the oxygen off, the hydrogen ions going this way, the electrons bouncing this way, electron transport chain is going to build this NADPH with this one hydrogen, ready to add energy where we need it there. All right, let's see. Let's ask some questions here. Which complex consumes hot water? It turns out this guy right here. We're going this way. So PS2 is the one that is involved with this whole breaking down of the water into two. What is added to NADP plus to convert it into NADPH? That hydrogen right there. Boom. Oops. Oh, I hit it right there. It goes in there and it hooks on there. And now we have this energized molecule ready to do its job. So let's look at this. ATP synthase drives energy from the generation of ATP from the movement of hydrogen ions down their concentration gradient from the thylakoid space to the stroma. What the heck does that mean? All right, here we got the thylakoid space right here. And so we have this increase of hydrogen ions that goes here, turns that thing ATP synthase complex, which I kind of call a turbine, and it drives that, and now we have energized this ATP and it's ready to do its job over here. So we got the two parts, electron transport chain making this guy, we've got the ATP synthase making ATP. Got it? Alrighty, oops, it's cut off at the bottom. So what's the order of this? It starts with the water. In what direction do the electrons of this circuit flow? Let's say, if we're thinking of this like a battery. It starts with water, then it goes to the PS2, then it goes to electron transport chain, and then it goes to PS1, then another electron transport chain, and NADPH. Boom. All right? There's a question on a test like that, just so you know. So you can actually see how this flows through right there. All right, now we've captured our energy and we're ready to use it in the Calvin cycle. So let's talk about that. It's powered by that ATP and NADPH we just generated, occurs in the stroma, and what are we trying to build? We are trying to build a carbohydrate. In this case, it's glucose. There's three main steps. First, we have to fix the carbon dioxide, then we reduce it, and then we make whatever the stuff that we need to start all over again. So it's a full cycle. Let's look at those more specifically. All right, fixation of carbon dioxide. All right, 6 carbon molecules split into 2 3 carbons. That's all that's happening there. So we have that glucose, or no, excuse me, we have a 6 carbon molecule and now we split it into 2 3 carbon ones. I suddenly switched into respiration on you there for a second. All right, then we have the reduction of the carbon dioxide, and it uses NADPH and some ATP. We'll see a diagram that shows this really, really, really well. Then regenerate what we start out with. So we have, we end up with 6 G3Ps, and 1's going to turn into glucose or something else, and 5 have to go back and start the whole thing over again. All right, let's look at it here. So here we fixate the carbon dioxide. See, this section is called fixation. This is the guy that we're going to regenerate and starts it all over again. So we're kind of hooking the carbon on that is what we're doing. All right. Then we reduce it. It takes energy. So we've reduced it. Then, oh, this is more of that. So we've reducing it. I put that in twice, I see. So we end up with 6 G3Ps right here. One of them is going to become glucose. That's all. All of this is to make that one G3P. The rest of these are going to go over and make that guy again. There you go. And whoa, we need energy to do it. So we've used ATP over here, NADPH over here, and ATP here again to start with our original stuff. So what are the purpose of the last 3 ATP molecules utilized in this cycle? Whoa, to turn this into that. So we end up regenerating our starter molecule, so this is a whole cycle and it starts again by adding that 3 ATP right there. We want to turn this into that. That's why. Which stages require ATP? Look at your diagram. All right, let's see. We need 6 over here for CO2 reduction. And we need 3 over here for regeneration of RUBP. Okay? Just learn how to read these things because there's no reason to memorize this stuff. Look at it and read it. Okay, so we got a problem. A poison disrupts the hydrogen ion gradient. What is going to happen here? What does the hydrogen ion gradient produce? Do you remember? Over here, check it out. Hydrogen ion gradient, boom, ATP. See it right there? So, if there's no ATP, what will build up? And where does it stop in the Calvin cycle? Right here. You see that 3PG? Right there. Because we don't have this ATP coming in because of this whole problem. No more ATP, boom, it stops right there because it can't go to that next step. All right, how many times must the Calvin cycle turn to yield a single molecule of G3P? All righty, look at your diagram. Net gain, 1 G3P. So it has to go around one whole time to make 1 G3P. Remember, 5 of them come up and become our starter again. So, how many times must the Calvin cycle turn to yield a single molecule of glucose? Well, glucose, it has 6 carbons. Guess what? It's going to have to go around there, boom, 6 times to make it. Crazy. What would you say has more chemical energy? These are all questions on the test, so I'm kind of helping you kind of think through and learn how to read these diagrams. So, do you think CO2 does right here, or GP3 right here? Sorry, it's down at the bottom. What our product's going to be? Well, if you think about it, we're going to build this guy out of some other things but CO2. So, more energy is going to be stored in this final product than in our building components. Make sense? All right. Another question. Chlorophyll is to the light reaction as blank is to the Calvin cycle. Let's think about that. So chlorophyll accepts the input, which is solar energy, final set energy in the form of electrons into making ATP and NADPH. And in the process, more chlorophyll is regenerated. Does that sound familiar? RUBP accepts input of CO2 and energy from the light reaction should produce the end product of the Calvin cycle, which is a precursor to a carbohydrate, G3P. All right, so RUBP is what we're talking. Watch this. All right, what's going to happen to that G3P? We're closing on the end of this. If you're getting a little overwhelmed, hang in there. We're almost done. All right, so we have that 1 G3P coming out of here. It can go this way and become an amino acid, glycerol, and fatty acids, which turns into a lipid right there. Or it can go this way and become these things. First, it goes into glucose phosphate. It becomes that first. Then it becomes starch or cellulose, different process, or sucrose. Alrighty? Plants and algae can make any molecule they need from this guy right here. Pretty amazing. So we've got other types of photosynthesis. We're finally out of that stuff. We get to talk about practical things now. So plants are adapted to the light and rainfall of wherever they are. That makes sense, right? So when light and rainfall are moderate, we have C3 plants doing their whole thing. That's what you're mostly familiar with, C3 plants. They're really successful in moderate temperature, lots of water, rainforest kind of conditions. They don't close their little stomata during the daytime because they don't need to worry about losing water. They don't do anything particular with that. Okay, let's talk about C3 plants a bit more. This stage, carbon dioxide, is fixation occurs in C3 plants by taking this molecule right here, combined with CO2, and it breaks it into 2 3-carbon compounds. That's the first step of it. But if the weather is really hot and dry, preventing water loss becomes very, very critical. So guess what? Some great little organisms, some great plants have evolved to meet those needs. Number one, they'll close stomata, that little opening on the bottom of the leaf, to limit water loss. It also limits CO2 intake and allows O2 to build up. So there's some issues kind of going on here. But we definitely are closing that down so the water's not going to evaporate from inside our plant. So these kind of plants are C4 plants. They use C4 photosynthesis. And in this situation, after that CO2 fixation, a C4 compound is formed instead of a C3. So let's look at it. How are they different? So the C3 plant has mesophyll cells arranged in parallel rows. You'll see that picture in a minute. And this guy is pretty different. It has a layered arrangement around leaf veins, and it has this shield bundle cells. That's to protect it from buildup of O2. Remember, that's going to happen when you shut down that bottom little opening. So it's the chloroplasts in those bundle sheath cells that are carrying out the Calvin cycle. This all is happening because there's partitioning of pathways in both of those, both carbon fixation and the Calvin cycle in the C4 plant, because it works better if it's kept separate. Now let's look at a picture of it. All right, this top one is the kind of plant you're very, very familiar with. These guys are the ones that are more adapted to hot weather, dry weather. This is that bundle I told you about. Bundle sheath cells. It's going to shield that RUBP from oxygen because it's going to build up because this little stomata down here is going to shut to avoid water loss right there. All right. So, C3 plants carry out both reactions in the mesophyll, which is perfect in moderate conditions, like we're, in Kansas when it's not too crazy hot and dry. In a rainforest, perfect for it. So C4 plants had to partition things off so the stomata can stay closed, conserving water, and they won't have problems with that oxygen buildup exposure to this rubisco. And it's a huge advantage in dry, hot weather. So here we go. We come in, it turns into a C4. It's in the bundle sheath. Remember I told you? That's where this is going to happen right here. Same thing's happening in here in a C4 plant. Oh, and corn is one, by the way. Then we can get even hotter and drier environments, and those are called CAMs, C-A-M. So during the night, CAM plants open the stomata when it's cooler, and they use C3 molecules to fix, forming C4 molecules, and then they store the C4 molecules. During the day, they keep the stomata closed to avoid water loss. And here's what they do. Pretty similar. Goes through that C4 thing going on again. And here an example is pineapple. So, evolutionary trends. We're almost done. You can cheer now. C4 plants most likely evolved in and are adapted to that area of high light, high temperature, and limited rainfall. They're very sensitive to cold. So C3 plants, the kind that aren't specialized here, they do better in colder areas. CAM do extremely well when it's really arid and dry. So it's very widespread. Okay, these C4 and C3 plants sound pretty awesome, right? Any drawbacks? Yes. Even though they can function more efficiently in hot and dry climates, they also produce a lower GP3 yield. They don't produce as much. Why? Because they need energy to move fixed carbon. Oops, typo, sorry. I typed that this morning. Fixed carbon into bundle sheath cells. And so remember, they're going to do that in that bundle sheath to keep it separate. And it takes energy to move it there, so they can't use that energy to make more GP3. Only drawback. And you are done! Yay!